Assalamu alaikum, my dear students of Biochemistry for Dentistry. Today, inshallah, we are going to have a new topic which is about paper chromatographic separation of amino acids. We explained the theoretical part of this experiment in the practical online session. We had it through WebEx, and I'm going to give you a quick revision before we start the practical part. The idea of the paper chromatographic separation, it's summarized by having a paper which is made of cellulose. We call it chromatographic paper, and this paper, on the bottom part we will put our sample and the standard chemicals. Then we are going to place this paper inside a jar. We call it a chromatographic jar, and in the bottom part of that jar we have a liquid. The paper is fixed in position. That's why we are going to call it as a stationary phase, and it is made up of cellulose, as we said, so it have high polarity. While the mobile phase is present in the bottom part of the jar, and it could move up over the paper. That's why we will call it mobile phase, and that mobile phase have low polarity. So whatever the spots we are going to put on the basal line or the basal part of the chromatographic paper is going to be moved up, but this movement will be judged by the polarity of the chemical compounds. If the polarity of the chemical compound is high, it means they will be adhered more to the paper, so they will not be moved for a longer distance. While if the spots having low polarity, it means they are going to be dissolved by the mobile phase, and it's going to be for higher distance. So if I have a mixture of two compounds, these two compounds, one of them having high polarity and the other having low polarity, the low polarity compound will move up for longer distance, while the high polarity will stick to the paper and will move only for smaller distance. Okay, so now we are going to find out how to do the practical of this experiment. Let's go to page number 2. I hope everything is clear for you and the camera is shooting everything. Okay, so now we have to prepare the standard solution, because here we have a powder. This powder made of a lot of amino acids, but I would like to make sure if it contains glycine or tyrosine or not. So I'm concerned only about the tyrosine and the glycine. So what should I do? I have to prepare standard solution of tyrosine, and I have to prepare a standard solution of glycine. Then I will compare the movement of the spots between the sample and between the glycine and tyrosine solution. So we have to prepare glycine solution and we have to prepare tyrosine solution. Both of them, the concentration is 1%, and even the sample solution, all of them are 1%. So how we are going to do that? Simply, we know that 1%, it means 1 gram in 100 ml. But we don't want to prepare 100 ml. We don't want to waste our standard. So we will use the 25 ml jar. So this 25 ml will require only X number of grams. So how we are going to do it? Cross multiplication, multiply 1 by 25 and subdivided by 100. So we will take 0.25 grams of tyrosine and place it here, and then 0.25 of glycine, and same for tyrosine and same for the sample solution. Okay, now we are going to prepare the mobile phase. Okay, so how to prepare the mobile phase? The mobile phase composed of 3 chemicals. One of them is the distilled water, the other chemical is N-butanol, and the third chemical is glacial acetic acid, as you can see here. The ratio between them is 2 to 4 to 3. It means 2 parts of distilled water, 4 parts of N-butanol, and 3 parts of glacial acetic acid. Okay, how to get that? I'm going to ask you to prepare 25 ml of that mixture. How to prepare it? Okay, first of all, you will say for the distilled water, the portion of distilled water, it is only 2 parts. We will subdivide it by the total number of parts, which is 2 plus 4 plus 3, which is 9. Then we are going to multiply it by the total volume of the solution we want to prepare, which is 25 ml. So we will do it by the calculation, by the calculator, I'm sorry, subdivided by 9, and then multiplied by 25. 2 by 9, multiplied by 25. So it is 5.5 ml. So the volume of water I'm going to use is 5.5 ml. Okay. Now we want to know the volume of N-butanol, the second chemical, so the N-butanol. the same technique. We will find out what is the number of parts of butanol. It is 4, subdivided by the total number of parts, which is 9, and multiplied by 25. So it's 4 subdivided by 9 and multiplied by 25. So it is 11.11 milliliter of N-butanol. Now we are going to detect the total volume of glacial acetic acid. So I will say glacial acetic acid. The number of parts of glacial acetic acid, it is 3. We are going to subdivide it by 9 and multiply by 25 ml, the total volume of the solution. So 3 subdivided by 9 multiplied by 25. So it is 8.3 milliliter. Of course, if we added all of them, 5.5 plus 11.11 plus 8.3, the total volume would be around 25 ml, which is the total volume we require to prepare from the beginning. Okay, so this is how we prepare the mobile phase or the mixture of the mobile phase. Of course, we have to use the measuring cylinder in that procedure. Now we are going to proceed with further preparation. Preparation of chromatography. How to prepare the chromatogram. When we say chromatogram, it means the paper itself. We call it chromatogram. This chromatogram is made up of cellulose, as we said. So the width of the paper and the height of the paper will be totally dependent on the experiment itself. We said we are going to use the chromatographic tank or chromatographic jar. This chromatographic jar have certain height. So we are going to measure that height by the ruler and we'll find out that it is 11 centimeter. So we will say that we cannot place this paper inside the chromatographic jar. So we have to cut it into certain dimension. How we are going to specify these dimensions? Very simple. We will say this is our paper. So the height is judged by the chromatographic tank itself. So we said it is 11, so we will make it 11.1. Why we need 11.1? Because the extra 1 centimeter, we are going to bend it and adhere it to the bottom of the cover of the chromatographic jar, so the paper will still perpendicular on the base. It will not be tilted or it will not fall down. Next, we have to specify the width of that paper. How to specify the width of the paper? We are saying that we need how many standard? 1 and 2. And we have 1 sample. So the total number of spots is 3. If you are going to multiply 3 spots by 2, so 6. So 6 centimeter, this is the width of the paper. We have to draw this by using pencil only. Ink is not allowed, so we use only the pencil. Okay, and we will draw this start line 1 centimeter from the bottom of the chromatogram or the chromatographic paper. How we are going to specify where are the spots? We will keep 1 centimeter from this dimension, and then we will base the first spot, then we will keep 2 centimeter, second spot, another 2 centimeter, 2 centimeter, and then 1 centimeter between the last spot and the wall. So to make it wider for you, I will explain it here. So here I will draw the baseline. Then from this margin to the first spot, I have to keep 1 centimeter. From the first spot to the second spot, I have to keep 2 centimeter. From the second spot to the third spot, again 2 centimeter. And then from the third spot to the wall, we have to keep only 1 centimeter. So if you count them, it would be 1 plus 2 plus 2 plus 1, which is 6 centimeters. So this is how we prepare the chromatogram. Of course, when we cut the edges of the chromatogram, it's not allowed to just tear it with the ruler. We have to use the scissor to ensure the uniformity of the margins. Okay. Later, how we are going to spot the chromatogram, we will simply take from the solution we prepared and we will take the sample using the capillary tube. This is the capillary tube we are going to use. Okay, then development. What is the meaning of development? Development, it means the movement of the mobile phase over the stationary phase. So when the mobile phase starts to climb up over that paper, so we will call this step as development. Okay, then after the development, we are going to localize the spot by something we call it chromogen. And then we are going to start analysis by calculation of what we call retardation factor. And then we will write down the results. So that would be all. Now we are going to move to the practical part of the experiment, so please stay with me. Before proceeding with this experimental session, we have to make sure that we have all the chemicals and instruments available at our disposal. Here we have the labels, ruler, pencil, scissor, chromatographic paper, chromatographic tank, 3 containers of total capacity of 25 ml, measuring cylinder of total capacity of 25 ml, capillary tube, spatula, weighing paper, top band balance. And for the chemicals, this is the glycine, tyrosine powder, glacial acetic acid solution, N-butanol solution, ninhydrine reagent in sprayer, and distilled water in the distilled water dispenser. Proceeding using top band balance, we have to make sure that the balance is leveled. So we have to adjust the level and make the bubble in the middle of the circle. So this is how we're going to do that, by adjusting the legs of the balance. So now it's leveled in the middle of the circle. So now we can proceed using the top band balance. Now we are going to use the top band balance to weight the 0.25 grams we needed to prepare the sample solution and the standard solution. Before that, we have to make sure that the top band plate of the top band balance is clean, so we can simply clean it using the brush. Then we will press on, and then we will press on tear. Now the reading is 0. So now we are going to put this weighing paper on the top of the top band balance. And then once again, we are going to press on tear. So we will neglect whatever the weight on the top of that balance. Now we are going to prepare the glycine solution. So we will take the spatula and we will take 0.25 grams of the glycine powder. And gently we are going to add that, and we will try our best to not to exceed the desired quantity. I think we are too close. You will notice that the sensitivity of that balance is much more lesser than the sensitivity of the analytical balance. So we are done with the glycine. Now we are going to transfer this amount into the container, and then we are going to add distilled water, we will make sure that we dissolve the quantity of the glycine inside. It's already soluble because it has a relatively high polarity comparing with others amino acids. Now we are going to add distilled water till the mark of the 25 ml. So now I have the glycine solution ready for me, and I will just write on that label here G as an abbreviation for glycine. So this is my glycine sample of 1%. Using the same technique, after preparation of glycine 1% sample solution, I prepared tyrosine 1% sample solution, and I prepared 1% of the unknown sample solution. We have it here. Okay, now we are going to proceed with that practical experiment. So this is our chromatogram or chromatographic paper, and we know that the width required is only 6 centimeters. So by the ruler, I will mark 6 centimeters for the width. And for the height, it is 12 centimeters. So here, or sorry, we said, yeah, 11 plus 1, so 12, yes. Okay, I think I have bad memory. Okay, and here once again, yeah, 6. So I think everything is just, we have just to tilt it a little bit to the right side. Then with the pencil, we have to draw very light line just as an indicator for the scissor later on, we are going to cut this paper. So here again. Now, using the scissor, I have to cut the paper, and I have to be as accurate as I can. Okay, so now this is my chromatographic paper ready for me. Now I will measure 1 centimeter from the bottom line, and then I will mark it here, and then again here to get a straight line. I will mark it here, then I will do a straight line. This is the line I'm going to draw right now, is the baseline. Why we call it baseline? It is exactly like the start line of the race. So we have to, we have to put all the spots on the same level, so when they develop, whatever the distance they will cross, it will indicate their similarity to the standard or the sample spots. So now we are going to equalize where we are going to put the spots. So we said 1 centimeter from the wall here. So here would be the first spot, and I will call this spot as the sample. So I will call it S. Okay. Now this is the second spot. We have to keep 2 centimeters. Okay. So here would be the second spot, and I will call it glycine spot. And here would be the fifth at 5 centimeters, so I have to keep 2 centimeters. So here it would be the tyrosine, and then 1 centimeter from that spot to the wall. So now we are ready. So the step we have done just right now, we call it the preparation of the chromatogram or the chromatographic paper. Now we are going to move to the next step, which is the preparation of the mobile phase. Now we are going to proceed with the second part, which is the preparation of the mobile phase. For that, we will need measuring cylinder and the solution which we are going to use to prepare the mobile phase, which is the acetic acid, the N-butanol, and the distilled water in the dispenser. Okay, how we are going to proceed that? It's very simple. I will take the measuring cylinder and I will add to that the total volume of water, which as we can see here, it is 5.5 milliliters. Then I'm going to add to that 11.11 milliliter of N-butanol and 8.3 milliliter of glacial acetic acid. So I will start by addition of glacial acetic acid, which is 8.3. So simply 8.3 plus 5.5, it would be 11.3. So I will fill up with glacial acetic acid till the mark of 13.8. Okay. Now we are going to add the N-butanol, 11.11 milliliter. So we have to add that to 5.5 plus 8.3 plus 11.11. So we are going to go to the mark of 25 using the N-butanol. Yeah. So we are done, and now the solution is ready of the mobile phase. So what we should do now, we should bring our chromatographic tank or chromatographic jar, and then we will place this solution inside the chromatographic jar or the chromatographic tank. So now the mobile phase is ready. Notice that the level of mobile phase is not exceeding 1 centimeter from the bottom of the chromatographic tank. That's why it will be very convenient for us when we place the chromatographic paper inside the tank, it will not exceed the start line or the baseline. Now we have to close the container well and keep it for a while to make sure that all the gases, all the volatile gases are saturated inside the container and the entire chamber is saturated with these gases. So we have just to simply place it here and keep it a while till we proceed with the next step. Next step, we are going to apply the spots from the different samples on the chromatographic paper. So we will get our chromatographic paper and we will get the capillary tube, and then we will use the capillary tube to take from the sample and the standard solutions. So we'll do it like this. Okay, this is glycine. So I will move perpendicularly and I will spot on the glycine. So this is the first spot. Now I'm going to take another capillary tube and I will take one drop from the tyrosine and I will spot it on the tyrosine here. And I will take new capillary tube and I will take from the sample solution and I will place it on the sample. Now I loaded the 3 spots. This step we call it loading, and I will dry the spot so I will get rid of any moist content. So I'll just have to do that till the spots becomes clear. And now I'm going to reload the spots again. So once again the glycine, I have to spot it on the glycine, and the tyrosine, I have to spot it here, and the sample I have to load it here. Now I'm going to dry it again. Now I'm going to use the cover to fix the chromatographic paper or the chromatogram on the lower part of that cover using the tape. So now I'm going to bend the apical part around 1 centimeter. If you remember when we said 11 plus 1, so this is the 1 extra. So I'm going to just bend it and I'm going to fix it here on the bottom of the cover of the chromatographic tank. Of course, there is many ways to fix the chromatographic paper in the chromatographic tank, but I think this is the most efficient way from my point of view, for sure. Okay, so now this is my chromatographic paper. It's ready, and I have just to place it inside the chromatographic tank. So I will open the chromatographic tank, and immediately I have to replace the cover, and I have to make sure that when I place it, it have to go down in perpendicular way. Now I'm going to observe the following development while it's occur. If you can see it, now the mobile phase is climbing up over the stationary phase slowly, and it's taking the spots up. Of course, the spots are not visual to us right now, so we have to apply certain chemical known as chromogen, and we are going to apply it later on to localize the locations of these spots. If you can notice that the level of the mobile phase is much below that the line of the start line or the baseline, and this is very important. Now we will allow it to develop. It will take around 30 to 40 minutes for the full development, and the solution should reach up to here, which is 1 centimeter below the top part or the apical part of the chromatographic paper or the chromatogram. Now 37 minutes already passed, so we will find that the solvent moved up over the stationary phase and reached to that level. So now we are going to remove the chromatographic paper or the chromatogram from the jar, and we will start to define the solvent front, which is the line between the dry and wet area, if you can see it here. Of course, we are going to mark that using the pencil. Now we marked the solvent front or the front line, and now we are going to dry that paper. Notice that there is no visual spots on. Okay, now we are going to dry the chromatogram. Notice here this is the pencil line, which represents the solvent front, the line between the dry and wet area. And now we are going to dry this chromatogram. Now, after complete dryness of the chromatogram, we are going to apply our chromogen, which is ninhydrine solution, and we are going to do that inside the fume cupboards. So now we are going to use the fume cupboards to apply the ninhydrine spray over the chromatographic paper after the development. So now we are going to turn on the lights and the suction force. Then we have to place our hands right here to make sure that there is sufficient suction force to take out any toxic gases are inside the fume cupboard. So now we are going to proceed with the next step, which is spraying the chromatographic paper after being dried by the ninhydrine solution. So this is the chromatographic paper. I will place it here just to support, as a support. Then I'm going to spray. Please make sure that the suction is working, so whatever the gas is coming from the ninhydrine sprayer, you will not inhale it. Now I'm going to spray it, as you can see. The function of the ninhydrine, it will react with whatever amino acid or protein sample are within this space of the chromatogram, and then it will colorize it into blue. But for sure we have later step, which is heating the chromatographic paper using the oven. So stay with me. So now we are going to use the oven, and the temperature is set at 110 degrees Celsius. So here we are going to place the support and the chromatographic paper, and we will keep it inside for 5 minutes. Now we are going to remove the chromatographic paper after the heating. So now we can see the spots starts to appear on the chromatographic paper or the chromatogram. Now we are going to take it and start the analysis. Okay, so this is the result we got after the ninhydrine application, which is the chromogen, and now we are going to understand what is the meaning of these colors. If you are going to look with me here, this is the spots of the sample. It gives us 2 different spots. And this is the spot of the standard glycine. It gives us only 1 spot. And this is the spot of tyrosine, give us 1 spot. Let's start by the standard spots of glycine and tyrosine, which we prepared at the concentration of 1%. You will find that the glycine traveled for shorter distance, while the tyrosine traveled for longer distance. Okay, what is the reason of that? Tyrosine having very low polarity, so it will be more soluble with the mobile phase. So when the mobile phase moves up, the tyrosine will be moving with the mobile phase for longer distance, while the glycine having high polarity, so it's kind of stick to the stationary phase. That's why it will not travel for a higher distance with the mobile phase. Okay, so for the sample we found here 1 spot and another spot. If we are going to look at this spot, we will find it almost parallel to this spot. So we will say that the sample, it contains glycine. And the same for this spot, which resulted from the sample spot. It's parallel to this spot, so we will say that the sample is containing tyrosine. So the sample is containing both glycine and tyrosine. So this is the qualitative analysis of the chromatogram. Now we are going to quantitative analysis of chromatogram. One more thing you have to note that this circle here represents the concentration of 1%, while this circle here represents again the glycine, but with unknown concentration. Okay, so can we use chromatogram in quantitative analysis? Yes, we can. How we are going to do that? Simply, we will find out the diameter of that circle, and we will find out the diameter of that circle. And then we will say, let's say the diameter of that one was 1 centimeter. So we will say 1% will give us a circle of 1 centimeter diameter. If we're going to measure the diameter of that circle, let's say it would be 0.25, the diameter, it would be 0.25 centimeter. So what would be the concentration? By cross multiplication, we will find out the concentration is 20 or 0.25%. Of course, this is not a very accurate way to get the concentration, but we can get a relative concentration. We can say that the content of glycine inside the sample having much lower concentration than 1%, because we are going to compare it with the standard glycine. And the same for the tyrosine. It's the circle here, or the spot here. It have much lower diameter than the standard tyrosine. So we will say again the contained amount of tyrosine, or the contained concentration of tyrosine inside the sample, it have lower concentration than 1% of the standard spot. One more factor we have to know that, which is retardation factor, and there is another factor which is retardation fraction. For retardation factor, how we can calculate it? It's very easy. We will get a ruler, and then we are going to find out what is the distance traveled by the spot. Let's name these spots into spot A, and this one we will call it spot B, and this one will be spot C, and this one will be spot D. So for spot A, this is the center of spot A. So we are going to measure the distance traveled from the start line to that spot. Okay, so I'm going to use the ruler in the right orientation. So it would be around 4.2. So for spot A, it's 4.2. Okay, and let's calculate it for the overall distance traveled by the solvent front, which is the distance from the start line till the solvent front. Let's measure it. It would be from here to there, which is 10 centimeters. So it would be over 10 centimeters. So the retardation factor we calculated by subdividing the distance traveled by the origin to the center of the spot, subdivided by the distance from the origin to the solvent front. So it would be the distance from here to there. So this is the distance, and then the distance from here to there. And we will calculate it here using the calculator. So it would be 4.2 subdivided by 10, which is 0.42. Now we are going to calculate it for spot C. This is the center of spot C. Again, we are going to calculate it from here to there. So it is around 4.2, 4.3. This is for spot C. And we are going to subdivide it by the solvent front, okay, which is from here to there, which is again 10 centimeters. So it would be 0.43. Notice that these two readings are very close to each other. Even if we subdivided them, we will get 0.42 subdivided by 0.43. It will equal to 0.976. This number is very close to 1. So we will say that spot A and spot C, they are identical to each other. Now we are going to do the same with spot B and spot D. For spot B, the distance traveled from here to there, it is 8.5. So this is for spot B, 8.5. And the total distance traveled from the solvent front till the, from the baseline to the solvent front, it's 10 centimeter. So it would be 8.5. And then for spot D, which is this one, we are again have to calculate it from here to the center, which is, I think, 8.678. So 8.8 subdivided by 10, which is from here to there, which is equal to 8.8. Once again, we can calculate a factor, and I will let you know what is that factor. We already done, and we are going to do it here again. 8.8. So it would be 8.8 subdivided by, 8.5 subdivided by 8.5 subdivided by 8.8. So it is 0.965, which is 6. Once again, you will notice that this number is very close to 1. This calculation, we call it a retardation fraction, and this again we will call it a retardation fraction. while this calculation we call it retardation factor for spot A and retardation factor for spot C, and the same retardation factor for spot B, and the same retardation for spot D. So now we know the retardation factors and retardation fraction, and we can use all these digits to define or identify the spots and get idea about what is the nature of these spots, even if they are glycine or tyrosine or any other amino acids. So this type of chromatography, we call it as ascending chromatography because the mobile phase used to ascend over the chromatographic paper. And we have many other types of chromatographic analysis, but this is the very basic idea of the chromatographic analysis. So by reaching to this point, now we achieved the aim of this experiment, which is chromatographic analysis of amino acids. Okay, now let's summarize what we had. We had one unknown sample, which we would like to know if it is containing glycine or tyrosine or any of them. So we have done through the chromatographic analysis procedure, and we got this result, which indicate that the sample contained glycine and contained tyrosine at the same time. So that would be all for today's experiment. Hope you enjoyed it, and see you inshallah in next session. Assalamu alaikum.